Today, students will be on our second topic for midterms, which we will be talking about proteins. Proteins are short chains of amino acids, and you know that your amino acids are the building blocks of our proteins. Amino acids in a peptide are connected to one another in a sequence of bonds called your peptide bonds. So between your valine, for example, and your valine and your leucine, for example, the one that connects these three amino acids is what you call your peptide bonds. Proteins, we have approximately 20% of which that comprises the substances in the body. They function as messengers such as your hormones, your enzymes, your connectors such as your collagen, your matrix protein, enzymes, you have your famous SGPT, your SGOT, you have your 5' nucleotidase, you have your, for example, LDH, lactate dehydrogenase, you have your integral structures such as your keratin, same, your collagen, and then transporters such as your, you have your ligands, your messengers are also transporters. So this an example of this is your Janus kinases, your JAK pathway that is protein mediated. Basic structures of proteins, we classify them into four: your primary, your secondary, and your tertiary, and your quaternary structure. Your primary structure comprises the sequence of amino acids in a polypeptide chain. The basic sequences in between makes your primary structure. It is the sequence of protein determined by the DNA of the gene that encodes the protein, or the one that encodes the portion of a protein for multi-subunit proteins. Just remember that your primary structure is a sequence of amino acids alone. Next, your secondary structure starts to fold of short, containing about 3 to 30 residues, contiguous segments of polypeptide into a geometrically ordered unit or units. The most common types of secondary structures are alpha helixes and your beta pleated sheets. Alpha helix resembles that of a coiled ribbon, and the carbonyl, your C double bond O, of one amino acid is hydrogen bound to the amino hydrogen, or the NH, of an amino acid that is 4 down the chain. Your beta pleated sheet are 2 or more segments of a polypeptide chain line up next to each other, forming a sheet-like structure held together by hydrogen bonds. The strands of beta pleated sheets may be parallel or anti-parallel. If the sequence of bonds go together in the same direction, that is your parallel. If it goes anti-parallel, it means that the sequence of bonds is formed from top to bottom and the other one is bottom to top, or that could be left to right, right to left, and left to right, and vice versa. An example of which is here, your alpha helix forms a helical structure which shows like a coil. Your beta pleated sheet, amo na siyang gina-fold. You can see here that it's like pleated. Hindi siya like your alpha helix that is coiled towards itself. This one is pleated. It means that there is just the turning around itself once. Your tertiary structure is the assembly of secondary structural units. So, your secondary plus secondary equals your tertiary. into larger functional units such as that of a mature polypeptide. The overall three-dimensional structure of a polypeptide is your tertiary structure. A special type of covalent bond that can contribute to the tertiary structure is the disulfide bond. So between your secondary structure is a disulfide bond promoting or allowing the attachment of your two secondary structures to form your bigger tertiary structure, which is three-dimensional in conformation. Look at this one. Here you can see that there are complexes of coils there, for example, and then plus another coil, and then this coil would coil around itself, which makes a 3D structure of your protein. So there is a disulfide linkage here, wherein it is bound by two sulfide bonds, or two sulfide or sulfur atoms. Last, the quaternary structure is the number and type of polypeptides of oligomeric proteins and their spatial arrangements. Proteins are made up of multiple polypeptide chains, also known as subunits. So one tertiary plus one tertiary equals to two tertiary structures. For example, like that coil siya, diba, on itself. Let me make this one another color. For example, this is your second tertiary. So this is your second. And this one is your first. Oops, sorry. This one is your first. So the combination of both makes your quaternary structure. An example of which is hemoglobin, which carries oxygen in the blood and is made up of 4 subunits, 2 of alpha and 2 beta types. So this is the structure of your proteins, the summary of which your primary protein structure is the sequence of amino acids. Simple as that. Your secondary protein structure, hydrogen bonding of the peptide backbone, causes the amino acids to fold into a repeating pattern. If it is pleated sheets, wala siya ga-coil on itself, but your alpha helix coils around itself. Next, your tertiary protein is a three-dimensional folding pattern of a protein due to side chain interactions. An example of a side chain interaction is, imagine this side chain here, and then the side chain of which nga ara sa likod niya. Kumbaga, for example, amo na siya, and then you have here another structure, diba? This is one structure, this one is in front, and this one's in the back. So, in between this one, the one in here, there is again your disulfide bonds. Just think of a magnet. Diba? This is one magnet and this is one magnet, positive, negative. There is that force of attraction that connects both. That is your disulfide bond. In the case of our proteins, that's your disulfide bond. Kaya kung magtapik na sila, there is a magnetic force, diba? That facilitates the magnetism. So that is like your disulfide bond. Hindi lang di siya kitaon because there is the three-dimensional structure of your protein. There is a 3D formation of your protein, which does not allow you to view from what is at the back. Next, let's go to protein denaturation. This topic is quite quick because this is just an introduction to proteins, and what we will be having more on is your enzymes, which we will be talking face to face next week. Denaturation, meaning the removal or without, right? Without naturation, or without its nature, without its nature or like its naturality. Okay, so this is the removal of the protein's nature. It is a process of modifying the molecular structure of a protein. It involves breaking down of many of the weak linkages or bonds within a protein molecule that are responsible for the highly ordered structure of the protein in its natural or native state. One measure or one method of denaturation would be by heating, which is the most common and the most cost effective. An increase in temperature will denature your proteins. Think of boiling pork meat. Di ba before the pork is boiled, before siya malaga, it is quite hard. It is budlay siya kumbaga do if the pork is underboiled, duga kaon ka bubble gum. It means that the protein structure of the pork is still compact enough that the proteins are adherent to each other, as compared when you boil it in water, that there are denaturations, there is alterations in the structure of the proteins, there is breaking down of some of the linkages, which makes your boiled pork meat softer. So may gingguba ka technically. Next is by treatment, usually by interference with bonding. When you use a strong alkali such as your NaOH, strong acid such as your hydrochloric acid, your urea, and your detergents. Your detergents are solubilizing, or they solubilize proteins by interfering with their bonds. That is why your soaps are effective germicidals, because a number of your structure of your bacteria or your microbes are of proteins. So if this one is denatured, if these ones are denatured by detergents, it renders them ineffective or non-invasive or even non-pathogenic at times. Vigorous shaking or biomechanical force is also one way of destroying proteins. For example, your centrifugation. Too much of centrifugation would lead to hemolysis. And we know that your RBC membranes has proteins like your integrins, your selectins, and you have your adherins. These are structural proteins. So if you apply vigorous shaking or vigorous agitation to that part or to that substance, for example, your red blood cells, that would lead to hemolysis because the proteins will be denatured or destroyed as per se. So that ends our topic for this week. Prepare a quiz for amino acids, proteins, and protein denaturation. Thank you.